So chapter 18 winds up our discussion here as we get started, and a lot of these are really the odd and the old. And so we talked about how there were three domains. And in chapter 16, we talked about viruses, which were considered acellular or non-living. And then we talked about archaea and bacteria in chapter 17. We're going to move on to the third domain, which is the eukaryotes. And so protists are a big group of eukaryotes divided into um, five categories or supergroups, if you will. And protists are so diverse. They're so diverse. They could be single cellular, multicellular. We could also talk about some of the weird ones. We could talk about single cellular ones that live in colonies, that live in groups. Volvox is a good example of that. That is an algae that works in colonies. So it's not really single cellular and it's not really multicellular. There's another one that is another algae or seaweed called Caulerpa. And this one again is super weird. So the cells of Caulerpa are huge. They're huge cells, and instead of making more cells, they just make more nuclei in their cells. So both of them are super weird. Take a look at Caulerpa on the internet. It is bizarre. And these are two examples of how bizarre and diverse the protists can be. So in their diversity, they get their nutrition in a lot of ways. We can have algae, which are kind of like plants, right? In that the fact that they are producers, they're autotrophic, and they get their light or their energy from the sun or light. We could also talk about protozoans being single-celled organisms that are chemoheterotrophic. So these are kind of like us, if you will. We're not protozoans, but we have similar energy processing. Or we could talk about fungus-like protists. And so in these ones, these would be animal-like, these would be plant-like, and these would be fungus-like. And so again, with those, they're like fungus in that they're chemoheterotrophic. And so that's how fungus works, and that's how we work as well. So as we take a look at these ones, we can go through, just how they are the same and where they came from in the first place. And so all protists, although they are diverse, they're all eukaryotes, which mean they have a membrane-bound chromosome or nucleus. We could have multiple chromosomes in here. And then we often have flagella or cilia with microtubules. So if we think back to chapter 4, that was an important thing that we talked about. And in chapter, what was that? 16, when I was talking about the extensions of bacteria, I said that the long extensions in bacteria didn't have microtubules, so they were analogous structures as opposed to homologous structures. Just as a little review here, and that's why I've put in the microtubules here. So these are the kinds of things that we learned about, like I said, in chapter 4. So some protists have a high level of cellular complexity, and there's so much variety. If there's anything to take away from this chapter, is that we're talking about a huge group, a huge class, and we are trying to classify them, but that is a daunting job. So as we talk about these, and this is again a bit of a throwback to chapter 4, we could talk about symbioses. So symbioses is a close association between organisms of two or more species. So as we get into ecology at the end of this course, we'll talk more about symbiotic relationships. But what we talked about back in, again, chapter 4 was the evolution of eukaryotic cells. And so that endosymbiotic hypothesis. And so this is the idea of how eukaryotes evolved from prokaryotes. And this evolutionary idea is also the idea that gives all of this diversity. And so these complex eukaryotic cells evolve when prokaryotes took up residence within larger prokaryotes. And so this was that endosymbiotic hypothesis that we talked about before. So in this next slide, if we take a look at this, this is one proposed explanation of how, say, a euglena, which is a protist that we'll talk about today, had evolved. And so over here, we have this primary eating, this primary endosymbiosis, where heterotrophic um, cell ate one that could photosynthesize. And so now all of a sudden we have producers, we have autotrophs. These are autotrophic eukaryotes. And then we get this secondary endosymbiosis. We get another eating where we end up with these heterotrophic eukaryotes. So these are consumers. And these heterotrophic eukaryotes. And so, this is only one protist here. You can imagine all the different variations of this, and over a huge amount of time as a theme when we talk about evolution, it's just almost impossible for me to stretch my brain long enough, big enough to think about the time that had to evolve here. So there is a tentative phylogeny, there is a taxonomy to these, there is a cladogram that shows the clades or the groups of protists, but is absolutely a work in progress. As I was getting ready to present this to the class, I realized that there was an error, that they have changed the cladogram that I had printed for the group, and between textbook publications, and I had some errors in the one that I was going to share with everybody. So it changes quickly, and there's also still discussion. There's not agreement on how things are organized. I also give it out. I think that memorizing and knowing the details of all this stuff is probably of less importance than just recognizing the diversity of the things that are on this cladogram and seeing the relationship of them. And so it's the relationship of these and the diversity that are really the big ideas when we're talking about protists and looking at a cladogram. So again, with these cladogram, it's the last page of this handout. I think I put it on the back of our review as well, and I've used it in, I think I even use it in our lab. But anyway, it's there because it's not something you need to memorize. I think that would be of little use, seeing as it's still a work in progress, and we're still trying to make sense of it. We are going to work through it in the fact that it's organized into these supergroups, and I think what's interesting about the supergroups is having an example that you may have some connection to, and so we'll take a look at them. As we work through the lab, we're also going to look at images of these. So I've included images as much as I could, but these are often much better than you would see under a microscope or in our lab. And so if you're doing the lab on your own, you'll be looking up images on the internet, but when we can see them in real life under the microscope, they often don't look quite as beautiful as some of these. So this is a supergroup that I have a hard time saying. Chroma, I can't say it. I'm going to pretend not to. I even made the internet play it for me a bunch of different times. So I'm not even going to pretend. If you're better than me, you can probably say it. So the first one are diatoms. So diatoms are single-celled autotrophic organisms with a silicate cell wall. And so there's a lot of silica in sand. Silica is also related to, so we could have a silicon-based life form. It's right next to or right below carbon on the periodic table. It bonds very similarly, and so it makes these molecules that are very similar, and they work with mostly aquatic environments and produce they're a key source of food in aquatic environments. So you may never have seen them before, but you may have heard of diatomaceous earth. So diatomaceous earth is often in filters. It's fossilized single-celled organisms, which are diatoms, and it's like a sand type material, and so it can be used in pool filters, in water filtration systems, sometimes in soil amendments. But diatomaceous earth has a lot of uses. And again, I just did a little bit of a deep dive on the internet. You can Google diatomaceous earth if you want to take a look at that. So again, the significance of these things and the really cool pictures of them as well. So the next ones that I would talk about are dinoflagellates. And so these ones have a little bit of everything in them. They are still unicellular, but they are autotrophs, heterotrophs, and mixotrophs. The diversity in each one of these groups is again so much. And so that's why we're not saying they're all autotrophs or they're all heterotrophs, because there's a lot of variety. Most of the dinoflagellates are, again, part of a marine and freshwater phytoplankton. And so phytoplankton would be little single-celled organisms. Some live with in coral organisms and coral reef communities. There's one specifically called Gymnodidium, and Gymnodidium is an organism that's associated with red tides. So I have not ever witnessed a red tide, but I do know that red tides can produce poisonous seafood. So clams and shellfish, where there is a bloom of Gymnodidium, can be poisonous or toxic to humans. And so that's an example of one dinoflagellate. Moving on in this supergroup, it is a huge supergroup. We could talk about an algae, brown algae. So brown algae would be some marine seaweeds or kelp. Kelp forests are significant in a marine environment. They're multicellular. They're autotrophic, meaning that they photosynthesize. And there is a beautiful picture of some kelp or seaweed. Another one here would be water molds. And so water molds can exist again in an aquatic environment. They can exist in freshwater as well. And they basically work on decomposing things. So this picture here is a water mold overtaking a dead insect, and it's going to break that insect down and help with nutrient cycling. Somehow my picture got over the end of my words. So these are unicellular heterotrophs. Again, they're going to take their energy from another organism, not from the sun. The last ones here would be ciliates, or these ones, and we've seen these ones before. So this is a picture of a stentor, but we have seen in other lessons a picture of a paramecium. They were the fuzzy little guys with lots of cilia on them. And so this is a common feature in ciliates, is these cilia. And so they can use the cilia to move or sweep food into their mouth. And so this stentor, we have, I know both stentors and paramecium in our lab. We have slides of those. And up here you can see the cilia on that stentor. So all rather diverse and rather beautiful. We've got single cellular, we've got multicellular, we've got autotrophic, heterotrophic, different environments that they live in, and this is all in one supergroup. The significance of these are huge. So diatoms, as an example, remember the diatomaceous earth and algae. All of these chromial I tried to say it and I couldn't get it out. I honestly practiced. Okay, so all of these, they're going to store food reserves in lipid droplets and carbohydrates. So this is important because they're a potential source of renewable energy. They don't need a lot of fertile soil. They can grow really, really fast. And so there's a lot of potential here for these being a really useful organism for us to use. We might use it in biofuels. And so because they produce these lipid droplets, we could harness these organisms to make biodiesel. I'm not saying we do it right now, but this is a potential use of these. The other one here would be fossilized diatoms, not just the diatomaceous earth. They also have been known to produce oil as reserves in the ground. And so lots of significance of these, and hopefully you can think of some connection to some of the examples that I came up with. The next supergroup would be Rhizaria, and so Rhizaria are amoebas of two different groups. And so Foraminifera, if we take a look at those, they have a carbonate test. And when they talk about a test, what they're talking about is like a skeleton, a skeletal-like structure. And so in these ones, you can kind of see what it would look like. They're quite beautiful to look at under the lab. I think we do have some foraminiferians in our lab to look at. Similar to these, but with a different molecule in their test or their skeleton, would be radiolarians. And so they would have a silicon or a silicate structure in their skeleton or in their test. And so they're both beautiful to look at. They both live in aquatic environments, and they're, yeah, pretty diverse and beautiful. Another group would be excavata, and so excavata have a feeding groove. Most of them have this ruffly type appearance here. So these would be red blood cells, human red blood cells. And this protist is, when present in the blood, can cause a disease called sleeping sickness. And so sleeping sickness isn't common in our world, but in tropical countries, sleeping sickness can be a problem. And so trypanosoma is the protist that can cause sleeping sickness. Similar, there is Giardia. Giardia is the protist that can cause beaver fever. It's a waterborne gastrointestinal beast, I guess. It's a horrible condition. If you've ever, you know, worked in forestry or done extensive backcountry travel, beaver fever or giardia is a worry for people, because it really does knock you down. It also needs to be treated. So a lot of these ones, again, cause diseases. And so sometimes that's how we interact with these protists, and they become important to humans. It's amazing what else might be out there that we don't notice or we don't understand. So there's another one here. So this organism causes a vaginal infection, a common sexually transmitted infection. And again, all of these have this commonality here of a feeding groove. And they also have a modified mitochondria where they can live without oxygen. One of the last groups that we could talk about are uniconts. Not to offend you, but you are a unicont. So if you take a look at your cladogram, unicont is down near the bottom, and you'll see that animals is the bottom entry on your cladogram. So if you can think about, the diversity of animals, each one of these things that ends our cladogram, there's that much diversity in these organisms. So when they say they're heterotrophic, autotrophic, and mixotrophic, that kind of makes sense because there's so much diversity. So the other thing here is uniconts is also a bit of a controversial group. Not that I have a problem with it, but that biologists haven't agreed on, how it groups and how it breaks apart. So there are amoebozoans in that group, and amoebozoans, this is an amoeba. If you've watched any of the Amoeba Sisters videos, they talk about how they named themselves the Amoeba Sisters, and this is an amoeba eating another cell, so eating a prokaryotic cell. The other one here, or sorry, not a prokaryote, another protist, another eukaryotic cell. The other one would be slime molds. And slime molds, you may have seen. So this is a dead tree where the bark has come off. And if you've ever seen this lace-like pattern, they can be bright in color. Those are slime molds. And so slime molds have different looks at different times in their life cycle, but this is something that's visible without a microscope, would be a slime mold. All of them have sort of this lobe shape pseudopodia, and so basically they have this amorphous shape where they can kind of ooze about in their environment. There's another kind of slime mold. Again, with this one's that kind of picture that we were talking about here. These would be like that picture that we had produced. The other thing here is that under adverse conditions, we get into some weird life cycles as we go. So the weird life cycles is that sometimes these slime molds can produce spores. A cellular slime mold can live as a solitary amoeboid cell, but when food is scarce, the cells can swarm together. Look at this cooperation of single-celled organisms, forming a slug-like aggregate there, and then it will form a long stalk at the top, and that fruiting body is going to produce spores. And so we're really starting to see some of these single-celled organisms work together, which is an important concept. It's an important thing to notice when we're looking at the diversity of these protists, because this is something truly wild and wacky, and it starts to make us think maybe a little bit more about plants or animals and cells working together. Um, the other ones that we could talk about here would be archaeoplastics. So archaeoplastics are red algae and green algae and land plants. So these ones are related because they do end up being some multicellular. Some of them are unicellular. Red algae specifically um, has carrageenan can be derived from red algae. Carrageenan is a food additive. Sometimes it's like more like a stabilizer or a thickener. And so red algae can be used for that. And this is an example of an archaeoplastics. It is a multicellular algae. Green algae could be in two different groups. You could have single cellular or multicellular. Um, there is an interesting single cellular green algae called Volvox that lives in colonies. And so these are single cells, but they live in colonies. So they sort of cooperate a little bit. The other thing that we'll talk about with green algae is that these multicellular green algae are the first to illustrate an alternation of generation. And so we talked about alternation of generation back when we talked about cell division, mitosis and meiosis. And this is a key characteristic of humans, animals, and land plants. So this is where algae and land plants start to show a bit of a connection here in this archaeoplastics. So algae is the closest relative of land plants. Like it says, it has this alternation of generations. There are two also really large, scary words, gametophyte and sporophyte. You're going to be able to see those in chapter 19. It's in the pre-reading to define those two. Basically, they're two different parts of a cell or two different parts of a life cycle where you have a haploid part and a diploid part. So humans have a haploid gamete and a diploid somatic cell, as an example, when we talked about mitosis and meiosis. In these ones, you know, this idea that we're not evolved from algae, or sorry, plants are not evolved from algae, but it shows a common ancestor. So there'd be several lineages that go on. And ultimately, you know, it took millions, or sorry, billions of years for multicellular life to arise. And so as we think about land plants and this connection between unicellular protists, the story of the evolution might look something like this. We're just thinking about it. So we get our unicellular protists, the idea of them forming a colony together, um, and then coming together with interdependent cells into, you know, a concept here that has a haploid and a diploid part of their life cycle, or alternation of generation. So these are big ideas, cells coming together, how things became multicellular, and also this alternation of generation. So the similarities between red or green algae and land plants, as they're part of the same group on the cladogram, is that they're multicellular, they're autotrophs, and they display this alternation of generations. The alternation of generation in algae would look something like this. You can see down at the bottom there is a key where the top part of my life cycle here is haploid. And in humans, that would be the gametes being produced. And then the rest of my life cycle is diploid. You can also see on here how we have meiosis, making cells go from diploid to haploid, and any reproduction within each side of the life cycle, it's always mitosis that's going on. And so we can see mitosis in a number of places here. Mitosis maintains the number of chromosomes, where meiosis cuts it in half, that idea of going from diploid to haploid. And the other important part, when we have meiosis and sexual reproduction, we'd have the fusion of gametes. So in humans, we call that fertilization. Okay, so moving on to fungi. Fungi, we're going to come back to those plants in chapter 19. Fungi are heterotrophic. They absorb their nutrients from their surroundings. Examples of these would be molds, other than the slime molds, yeast, penicillin, mushrooms. Most of the fungi consists of thread-like structures called hyphae that make up a mycelium. So a mycelium is like a mesh type um, mass of white thread-like things. In the garden, or if you're digging in the forest underneath leaves, you might find a mycelium of a fungi. We are going to talk about mushrooms, but the mushroom would have a mycelium attached to it. So these hyphae in the mycelium have a huge surface area, and they're going to absorb their nutrients from their surroundings. The hyphae is surrounded by a cell wall with chitin, and chitin is a structural carbohydrate. If you remember back from chapter 3 when we talked about those important polysaccharides, chitin was the structural carbohydrate, often in insects, but also in fungi. So this is just showing the difference. A whole mycelium is made up of many hyphae, and they're all cells with cell walls. And so this would be the bottom side of a leaf, if you can imagine. I'm making this video in the autumn, in the fall, and you know, the leaves are all falling, and in a few weeks, if you pick up a pile of leaves, you might see that white thread-like structure on the bottom of them as they're decaying, and the fungi is absorbing those nutrients. So fungi are hugely important. I love mushrooms. I would have to say that diet is something that's really, you know, close to my heart when I think about mushrooms. The other part is that they're really important in ecosystem. They're good decomposers that help cycle nutrients in our ecosystem. The other thing that we could think about is, you know, where medicine and biology come together, or medicine and nature come together. So there's many drugs that are derived from fungi, specifically antibiotics. You might have noticed that penicillin was in that list before. A lot of those antibiotics, like we had talked about, because of the peptidoglycan in bacteria, can act on prokaryotic cells, or bacterial cells specifically. Bioremediation is another cool idea or potential thing. So some fungi can actually break down toxic substances like DDT or cancer-causing chemicals. It's pretty amazing to use a mushroom to do that absorptive work and help cycle the nutrients in a safe way. Fungi are classified again, and we don't have a cladogram for fungi, thank goodness. If we think that protists are strange and odd, and we think plants have a further complexity to their reproductive alternation of generations and all that. Fungi are next level, and so they actually classify fungi with their reproductive structures. You know, it's just, again, the diversity on Earth is mind-boggling. So most mushrooms reproduce through spores, and if we were to talk about the connection between fungi and mushrooms, mushrooms are the fruiting body of fungi. They're the things that produce the spores. And so this is how new mycelium can grow, or new mushrooms can be formed. Mushrooms are just a part of the life cycle of fungi. The last little bit for us to chit chat about here are lichen, and lichen, again, bring to mind this idea of a symbiosis. So not endosymbiosis this time, or the endosymbiotic hypothesis, but it is a close connection between two different kinds of cells. And so there's a fungi and an algae living in this symbiotic arrangement. And basically each does their part. And so the fungi absorbs water and minerals and provides that to the algae. And the algae photosynthesizes and provides carbohydrates to the fungi. And so in this, they can help each other live in little to no soil. You can see lichen growing on rocks. They don't need soil. They don't need consistent moisture. They're amazing because of this the symbiotic hypothesis. So I said the wrong words. This symbiotic relationship. Whew, so many words. Good thing I got to the end without tripping over my tongue too much. So that does our chapter 18. 16, 17, and 18 are a bit of a unit for us and would be your next test.